Hello, I'm Ruthann Gorrell, and I've been working with Dr. Jonathan Lowry, who works out in Marian University College of Osteopathic Medicine. Dr. Lowry is interested in the cellular mechanisms behind osteoporosis. In particular, he's interested in the BMP2 pathway. BMP2, bone morphogenetic protein type 2, binds to BMPR2 and BMPR1A, so bone morphogenetic protein receptors type 2 and type 1A, in order to activate SMAD158, which induces bone matrix formation. In some of his recent studies, Dr. Lowry genetically knocked out BMPR2 in the skeletal progenitor cells in mice. What he found from doing this was that the mice that had the genetic knockout had an increase in bone density as opposed to the control group. In addition to this, there was a reduced age-related decline in bone mass as opposed to the control group. So this was a very exciting study, and the mechanisms behind this is being pursued in other work. In this study, we were interested in if there was translational potential here. So we sought out to identify commercially available antibodies that can block ligand binding in BMPR2. To do this, we used a cell-free immunoprecipitation assay quantified by ELISA. So we bought a kit for an ELISA, and we had wells that were coated with anti-BMP2. So we add BMP2 in, the BMP2 binds to the anti-BMP2, then we add a substrate on top that binds here. The substrate absorbs light. We can then measure the absorption of light. And so if we look here, as we add BMP2, we get an increase in absorption, which is exactly what we expected to see. So here we both validated the ELISA, as well as got an idea of how much BMP2 to use, so we are in a linear section of this data. So now that we had validated the BMP2 ELISA, we moved on to proof of principle for the ligand binding assay. To do this, we used noggin. Noggin is known to bind to BMP2. So if the noggin binds to the BMP2 can no longer bind to the anti-BMP2. And which is exactly what we found. So as we increase the concentration of noggin, we get a resulting decrease in signaling from the ELISA. So we now proved that noggin effectively inhibits BMP2, which proves the principle of our assay. So now we've got to move on to our protein of interest, BMPR2 ECD, or extracellular domain. So we add in BMP2, and the BMPR2 ECD binds to it. Now this is no longer free to bind to the anti-BMP2, just as we saw with noggin. And this also worked. As we increase concentration of BMPR2 ECD, we get a decrease in signaling from the BMP2. So this was all very good. We had so far we've just laid out what our assay is. Now we're finally getting to the point of our assay. Can we identify an antibody that blocks the BMPR2? And so we added 3 of 6, which is a mouse monoclonal antibody. The 3 of 6 bound to the BMPR2 ECD. With this blocked off, BMP2 can now bind to anti-BMP2. So now it can increase signaling. And so this worked. As we add 3 of 6, we get a resulting decrease in the activity of BMPR2 ECD. So this was really exciting. We were happy to find that this worked out. Since the 3 of 6 was prepared in an ascites preparation, we tested a second lot of 3 of 6 and found that it effectively inhibited BMPR2 ECD activity as well. In addition, we tested a control ascites preparation in order to make sure that the ascites had no effect on the experiment, and it did not. And then we tested 1F12, which is another mouse monoclonal antibody, and that also effectively inhibited BMPR2 ECD activity. So far we found 2 antibodies that effectively inhibit BMPR2 ECD, which is very exciting. However, we needed to know, is there more translational potential than just this? So we moved from a cell-free assay to cell-based assay. To do this, we used HEK293 T cells, or human embryonic kidney 293 transformed cells. And first we tested the cells to make sure that they had BMPR2. They did. So we were good to go. Now, if we move back down here to this pathway, we have BMP2 binds to BMPR2 and BMPR1A in order to activate SMAD158. So whenever SMAD158 is activated, it's phosphorylated, and we can measure for that protein in a Western blot. If the 3F6 or the 1F12 effectively works, it'll bind here and prevent phosphorylation here. So if we come back over here, we're going to test and see if we get phosphorylated SMAD158. So first we grew some cells and we didn't add any BMP2. Without BMP2, we shouldn't get any activation of the pathway, which is exactly what we saw here. When we quantify the proteins by Western blot, we have no phosphorylated SMAD158, which means we did not induce the pathway. So next we throw in BMP2. As you see in this second column here, we now have phosphorylated SMAD158, which means we've induced the BMP2 pathway. This is exactly what we've expected to see. So now we're finally getting to what we're interested in. Does the 3F6 effectively inhibit the BMPR2? As you see here in this final column, we have inhibited the BMP2 pathway by 3F6, which is very exciting. So this matches what we saw in our cell-free assay, that 3F6 inhibits BMPR2 binding. So since this was prepared in an ascites preparation, we did a test run with control ascites and found that it had no significant effect on the assay, which meant that the effect would have been the 3F6 and not the ascites. So this is very exciting. Everything has laid out very well here. We can't say definitively that the 3F6 has been blocking the BMPR2 here, but that's most likely we'll verify that by future experiments just to knock down BMPR2 expression. So pretty much what we have found here is that BMPR2 function can be blocked by neutralizing antibodies. We found in our cell-free assay that 3F6 and 1F12 mouse monoclonal antibodies can block BMPR2 ECD ligand binding. And then in our cell-based assay, we found that 3F6 can effectively inhibit the BMP2 pathway. We also had an incidental finding that BMPR2 is the major receptor for BMP2 and HEK293 T cells. So future direction for this, we will examine 3F6 and 1F12 neutralizing activity of BMPR2 ECD function in skeletal cells in vitro. And then if that works, we'll move on to systemic delivery of 3F6 and 1F12 in vivo in order to see if they regulate postnatal bone mass. Thank you so much for watching. We really appreciate it. And then feel free to contact us if you have any questions. Have a wonderful day.